So we're talking today about the role of non-coding RNAs in gene expression. So the last two videos also talked about gene expression, whether DNA is coiled tightly or not, whether the machinery like the RNA polymerase and transcription factors can get to the DNA or not, whether they're blocked. We talked about RNA processing with exons and introns. And we talked about this section too, but we're going to come back to this right now with RNA interference. So this is really just a continuation of the talk that we did last time. So I want to talk a little bit about RNA interference today. First of all, only a small fraction of DNA actually codes for messenger RNA and ribosomal RNA and transfer RNA. A lot of the genome, a lot of your DNA codes for this non-coding RNA. And there are a whole bunch of different types, and you don't need to memorize any of them. I just want you to get the gist of what they do. So non-coding RNAs regulate gene expression at a couple of points. One is at mRNA translation, so from here to here, and another is with chromatin configuration. So some of these messenger RNAs will bind to DNA and coil it up more tightly so that RNA polymerase can't get to them, and sometimes they will actually degrade the messenger RNA so it can't be translated. These are the different types of RNA. So you already know what messenger RNA is, and then you know what transfer RNA and ribosomal RNA are. We're going to talk about non-coding RNA today, and we'll talk a little bit about microRNAs and small interfering RNAs and piRNAs. You really do not need to memorize any of these. Just, again, get the flavor, the idea. Okay, RNA interference, the definition is the inhibition of gene expression by RNA molecules. So these are the non-coding RNA molecules that go back and stop gene expression in some way. The first way the scientists learned about them is they injected a cell with RNA and it actually decreased gene expression. So that's when they started to learn about it. Okay, one example is microRNAs, and they are actually transcribed in this long transcript of this long primary transcript. And then we have an enzyme with an adorable name. It's called dicer, and it will cut some of these like this. And then you'll degrade this side, and what you're left with is this small microRNA, and that binds with a protein. And then whenever this code, the AUUCA, whatever this code is, if that can bind with another piece of RNA, so this would be messenger RNA, and the microRNA would be the yellow piece. If they bind perfectly, that will tell the protein to break down that messenger RNA. So the messenger RNA is not going to get translated because it's broken down. If they code up pretty closely like this, but not exactly perfectly, it won't actually break down the messenger RNA. It'll just block a ribosome from attaching. Either way, it stops translation. The messenger RNA matches the microRNA and it's degraded or its translation is blocked. So that's what we just said. Small interfering RNAs are another type of RNA that do actually the same thing, but they're made from different precursors. It doesn't really matter if you know that part. Okay, transposons, these are actually insanely interesting. Another name for them is jumping genes or parasitic DNA. So you know how anything like, I don't know, a tortoise or a palm tree, anything that can make copies, that can live and make copies of itself is going to do so. And then the next generation and years and years and years from now, the world will be populated with the organisms that previously had been able to make copies of themselves, right? Like a meme. A transposon is really the same thing except at the genetic level. So a transposon is a piece of DNA that can copy itself and jump to another area. Not so good for the organism, but it does copy itself. So natural selection sort of selects for transposons at the molecular level, but maybe against them at the organism level. So when this does happen, it might go in the middle of a gene that you need. So here's a gene that you need, and the transposon hops up and interrupts it. Now you don't have this gene that you need, so that can actually be a big problem, and we have evolved ways to stop this from happening. Pwi-associated RNAs, these RNAs that we have DNA that codes for them, right? They can induce heterochromatin, so they can take a transposon and make it bind up tightly with its proteins and not get expressed with its histones. Sorry, that's the word I wanted. Okay, so RNA interference can happen at several levels. It can happen at the chromatin level, meaning they'll help to coil up the DNA and block it from getting transcribed. It can happen at the mRNA level, meaning if they bind together, like right here, then it can degrade the RNA. If they don't bind exactly perfectly, then it can just block translation. So these non-coding RNAs can regulate gene expression at all these different steps. The evolutionary significance of this, we don't really know, but it seems like the more complex organism you have, the more of these microRNAs you have. And so it looks like it's just another mechanism for us to regulate gene expression and have more fine control over it. So a really simple organism like this worm, C. elegans, doesn't have so many microRNAs, but the alpacas have a lot of them.